Hi, Dr. Biology here. This is an A-level biology exam paper commentary run through of the AS paper 2 from 2017. I've already done AS paper 1 if you want to check that one out. This is going to be very similar, obviously going through paper 2. I'll be talking about exam technique, things to look out for in the answers, and obviously I will go through the answers for each question. So I hope that will help with your revision for forthcoming mocks or exams. And yeah, so without further ado, let's get started. So this paper 2 is 1.5 hours long, and as you can see there are 9 questions. 8 of those are short answer questions and 1 of them is a comprehension question. So you get 75 marks, which is the same as paper 1, and the spec number is 7401/2. Question 1, and it talks about glycogen and cellulose are both carbohydrates, so that will directly be related to 3.1.2, which is about carbohydrates and specifically about polysaccharides. So it says describe 2 differences between the structure of a cellulose molecule and a glycogen molecule. So remember in your answer, because it's 2 differences, okay, you've got to mention both cellulose and glycogen in your answers. It's really important to state, and you have to do that for 1 mark. So the possible answers here, so cellulose is made of beta glucose and glycogen is made of alpha glucose. Second point, cellulose molecules have a straight chain and glycogen is branched. Thirdly, cellulose has straight chains and glycogen is coiled. And then the last one is about glycogen has a 1:4 and 1:6 glycosidic bond, and cellulose has only 1:4 glycosidic bonds. Notice it does say ignore references to hydrogen bonds, okay, and also microfibrils. So just wants you to talk about the structures, okay, so the basic structures that are different between the 2. This question is a nice start because it's saying describe, so that is basic recall, it's assessment objective 1, and if you've done your revision you should be able to do those kinds of questions quite easily. Question 1.2, it talks about starch. It's often stored in plant cells, and it's got 2 command words there, describe and explain 2 features of starch that make it a good storage molecule. So for this question you're just going to be talking about starch, but for 1 mark you need to describe. So describe the feature of starch and then explain why that makes it a good storage molecule, and that's just for 1 mark. So here's the mark scheme. You can see that as it says, it requires the feature and the explanation. There's a lot of answers you can have, and it says any 2 from the following. So insoluble, so it doesn't affect water potential. Branched, so it makes the molecule, or coiled because it makes the molecule compact. You can say branched, coiled, or alpha helix, so it can fit many in a small area. Polymer of alpha glucose, so it provides glucose for respiration. More branches, so therefore faster breakdown by enzymes, or it's large, so it can't cross the cell membrane. Could mention things like water potential, but you can't say insoluble, so it doesn't affect osmosis. So you've got to talk about water potential there. Ignore about surface area, do not allow reference to doesn't affect water leaving cells, but they would accept branched, so glucose is readily released. And then question 1.3 about food tests require practical, so looking at the test which would be used to show the presence of starch, so that is definitely required practical. And the one, well, it's not acid hydrolysis, it's not Benedict's, because that is related to sugars, okay, not starch, so it's like glucose monomers. Emulsion test is lipids, so it must be the iodine potassium iodide test. Question 1.4, and it says figure 1, which is a rather nice figure, I have to say. It's a rather nice photomicrograph, and it shows a section through a plant tissue at a magnification at times 500, tells you which is the starch grain, which is this thing, okay, and it says calculate the actual diameter of the starch grain between points A, which you can see there, and point B, which is there, okay. So here is my I am triangle, as I put it. So I want to find out the actual diameter, so I'm finding out the actual, so that will be image divided by magnification. So with my ruler I'm going to measure between A and B. I can tell you now that that is actually 20 millimeters on the exam paper, and you would use your ruler. There's a tolerance of plus or minus 0.5 millimeters, which is not a lot. So I would suggest you have a really good ruler. Maybe ditch the ruler that you've had since year 7 or in primary school. It's got bits jagged out of it, or the millimeters are fading away. I would make sure that you invest in a very good quality ruler, and then you can work out the answer. So we have 20 millimeters, well, we've got to convert it into micrometers, so times it by 1,000, so that equals 20,000, and then just divide it by the magnification, which is 500, and you'll get an answer of 40 micrometers. And then 1.5, it asks you what type of microscope was used to obtain the image. Give one piece of evidence. Well, let's have a look at the image, and what a fine image it is too. So you'll notice some elements of the image. Magnification, it's not all that higher magnification, so that would include a light microscope, but it's not a light microscope because you can actually see that there is 3 dimensions within that image. You can a 3-dimensional image, so it must be a scanning electron microscope. Notice in the mark scheme they will allow you to put SE or SEM. So question 2, and it says the cells of beetroot contain a red pigment, and they investigated the effect of temperature on the loss of red pigment from beetroot. So they put the discs into tubes containing water, and they maintained each tube at a different temperature. So different temperature, and then after 25 minutes measured the percentage of light passing through the water in each tube. Immediately you should be thinking, well, this is about transport across cell membranes, which is 3.2.3. But more importantly, you should know that it is required practical 4, which investigates the effect of a named variable on the permeability of cell surface membranes. So in this case, it regards temperature. So question 2.1, the student put the same volume of water in each tube. Explain why it was important that he controlled this experimental variable. Well, if you look at the stem of the question, which is my favourite pastime in these types of exam, you can see that it's you're looking at the effect of temperature on the loss of red pigment. Okay, so the red pigment from the beetroot. So if you're going to use different volumes of water, if there's too much water, the concentration of pigment will be lower. Okay, so therefore the solution will appear lighter. Or if there's too little water, the concentration of the pigment will be greater. If you have the same volume, why is it important then to control it? Well, very simply, it allows you to compare the results. So you're going to compare the results at different temperatures. With these types of questions, when you're looking at control variables, it's really important that looking at a factor, for example, such as the volume of water in this case, you want things to be the same so you can compare your results, because the independent variable is going to be the temperature, the thing that you are changing, and then you're measuring the red pigment probably using a colorimeter. But you need to know that that's quite a common style of question. So the first part is really explaining if the volume of water is different, well, what effect would it have, and then why? What effect would it have? Well, the results. If you don't keep it the same, the results won't be comparable. But if you do, which is what they want you to talk about having the same volume of water, then the results are comparable. Question 2.2, and it says describe a method the student could have used to monitor the temperature of the water in each tube. Now, obviously you would use a thermometer, but you wouldn't just say use a thermometer. You need to say that you would take readings, not just one reading, because it's saying you're monitoring the temperature in each test tube. So it's going to be take the readings using a thermometer. Now, if you said take a reading using a thermometer, that would be incorrect, because it does say you need to monitor the temperature. So that would assume that the water is checked regularly more than once during the course of the experiment. Sometimes when they're looking at required practicals, they sometimes give you data and they'll ask you to plot the points and then draw a suitable curve. But as you can see in this case, they've done the plotting for you and they want you to draw a suitable curve. So this graph is not a straight line. It's also not necessarily a curve as well, because you need to go from 20 degrees C, and you can't go just straight down like that because you're missing out the reading at what is it, 26. Okay, so I'll rub that out. So let's get rid of that. So it's actually an S-shaped curve. Oh dear, that's wrong as well. Why is that wrong, Dr. Burgess? Well, I have extrapolated below 20, which is incorrect. I have to start at 20. So I'm just going to magnify it so I can see it. Okay, there we go. Right, here we go. So here's my line. Oh, it's really bad. It should be an S-shaped curve. Okay, so not great, but that's the best I can do on this software. So a smooth S-shaped line of best fit, or because you can't really determine a suitable line of best fit in this data, you could get a ruler and literally mark it point to point with a straight line. That would be acceptable as well in this case. As I said, they would reject any extrapolations. So when I mean an extrapolation, you can't go that way, and you obviously can't go that way. So be careful not to extrapolate the line. Then question 2.4, and it says the decrease in the percentage of light passing through the water between 25 and 60 degrees C is caused by the release of the red pigment from cells of the beetroot. Suggest, that's the command word there, suggest how the increase in temperature of the water caused the release of the red pigment. That's 2 marks. So nowhere in the question does it mention the cell surface membrane. So that would be the first thing, is why is it being released? Well, there's damage to the cell membrane. Why is that the case? Well, usually it's when proteins denature, or as the temperature increases, you get increased fluidity or damage to the phospholipid bilayer. So there we go. So all in all, that question, again, if you know you require practical, you know scientific methodology, is not too bad at all. Okay, question 3, and it starts off with a lot of information in the stem of the question. And it talks about, it says, most human cells contain 2 copies of each gene. However, there might be up to 15 copies of the gene for amylase, called AMY1. Scientists investigated the number of copies of the AMY1 gene in individual people in 2 populations. One population had a high starch diet, and the other population had a low starch diet. Figure 3 shows the results, which we'll talk about in a second, but looking at the question, to show you the question, they want you to describe what the results show, and they're going to give you a whopping great big 3 marks for doing this. So let's have a look at the graph and let's talk about how to tackle this type of question. So the graph shows the percentage of individuals going that way, and then on the x-axis is the number of copies of the AMY1 gene per person. So let's look at high starch first. So you can see high starch, just joining the points together because I think it's easier to see, and then low starch, I'll use it as green. Okay, so low starch has a bigger peak, but then it decreases quickly. Actually, the 0. Well, the first thing I notice is that with low starch there are actually fewer copies. So you can see most of the copies are in this region around here, and actually there's fewer copies. There's only a few individuals with 13 copies of low starch, but that's about it. Whereas high starch, you can see there are more copies. Between both high starch and low starch, you can see that the ranges do overlap. So they're very much, look at the lines, there's overlapping, particularly from 2 to 13 copies. Now in the stem of the question, it does mention, if I just show you, most human cells contain 2 copies. So if you look at the 2 copies there, very few, and you could say 2 to 3% have only 2 copies. So that's quite surprising in this data, because most human cells contain 2 copies. And then let's look at the data. So the mode, or the highest percentage of individuals for low starch is at 4 copies. You can see 4 copies there, whereas for high starch it is 6 copies. And lastly, if you look at high starch, you can see, as I've shown you, the range or the spread is much greater compared to the low starch. So there's lots of different descriptions you can make. In your exam, you're very much allowed to write on it. It's your exam paper. You know, if you want to draw things, make little notes, that's fine, because the examiner, if I just show you, when the examiner gets your answers, all they will see is pretty much that. So they won't actually see the graph, they'll just see your answer. In fact, actually they'll probably see it like that. That's why they say always write within the margins. So there are all the possible answers. Now, I think it is a really good revision technique to actually find as many graphs as possible as you can and be able to describe what the results show. So you need to use data that they give you. You can talk about ranges, the mode, the median. You can talk about the spread of data. You need to use some of the, as I said, the data, so the numbers of copies are mentioned, the percentage of individuals. So all those types of things need to be included in your answer. Sorry, for this answer you only need 3 of these points to get the full 3 marks. Now the question is talking about producing genes. Okay, so the AMY1 genes, and it talks about it's an adaptation of high starch diet, and it does say use your knowledge of protein synthesis and enzyme action to explain the advantage of this adaptation. So this is related to 3.4.2. So all it's looking for, it gives you a massive amount of space here, but if you just look at the answer, it just wants you to say about the fact that there'll be more mRNA, which means more transcription. You could just say more mRNA, therefore more translation, and the enzymes, you can mention amylase if you wanted to for the enzyme, and so the enzyme hydrolyzes speeds up the reaction. Actually, sorry, it doesn't hydrolyze. So the enzymes the enzymes are present, so the reaction is faster. And then question 3.3, it gives you a bit of a curveball here, so it goes away from protein synthesis to talking about natural selection. So multiple copies as an adaptation to high starch diet again, but they want you to say, well, why is this, suggest how it's evolved from natural selection. So in terms of natural selection, what is producing these extra copies of the AMY1 gene? And very simply, it's due to mutation. So therefore, those with more copies of the AMY1 gene are likely to survive on this high starch diet. And if they survive, they pass on the AMY1 gene, multiple copies of the AMY1 gene to their offspring. So whilst you're talking about genes, this is natural selection. So mutation produces the extra copies. Those with the mutation, they reproduce or they survive better on the high starch diet, and they pass on those multiple copies to their offspring. Question 4 starts off with a graph, and you can see you've got standard deviation on that, and it says that the scientists investigated how the concentration of protein in blood plasma changes in people between the ages of 60 and 95. So, question, what is the difference between males and females in the fall in mean concentration of protein in blood plasma between 60 and 95 years? So not between 60 and to 95, 60 and 95. So for 60, age 60, you can see that that point there is 69, and that's 71, so that's 2. And then if we look over here at age 95, just reduce it so you can see the scales, you'll see that that is 60 and that one is 64, so that's 4. So 4 plus 2, and that will give us the answer, which in this case is 6 grams per dm cubed. There we go. Now, difficulty is that I think a lot of students probably, when they did this, they did all the years. So, well, not all the years, but they did every single point and looked at the differences and added them up. But it says 60 and 95 years. 4.2, and it says use figure 4 to calculate the rate of change of the mean concentration of protein in the blood plasma of males between the age of 60 and 95. So here's the graph, and we want to look at the clear diamonds here, so 60, so age 60 to 95. So the first thing to read is at 60, you'll notice that it's 69, okay, so 69, whereas 95 it's 64. So that's a difference of 5 divided by the number of years between 60 and 95, so that is 35 years, and you'll get your answer, which is 0.14. It's a negative number, it's minus 0.14, but you don't need to put the negative number. So that's how you would work out the rate of change. Question 4.3, it says, what can you conclude about the effect of aging on the mean concentration of protein in the blood plasma in males and females? Now it's important to state that in the graph they do show you the standard deviation bars. You need to know that when they are not overlapping, they are significant. So that's a key thing. So if you look at the graph, so if I just bring up the graph here, you can see that the only time that the standard deviation bars do not overlap is at the age of 95. So that's one of your answers, okay. There's a difference, it's only significant at 95 years because the standard deviation bars do not overlap. You can also see that protein content in both males and females decreases in both sexes. However, it wants to know about males and females. Well, it decreases more in females. So you need to say the protein content decreases with age and decreases more in females. So there we have the mark scheme answers. Notice that you can't just say the protein content decreases with age. You've got to mention and it decreases more in females to get the 1 mark. Question 4.4, and it talks about the measuring the absorption of the blood plasma using a colorimeter, and they used a calibration curve to find the concentrations. And then the question says describe how you could obtain data to produce a calibration curve and how they would use the curve to find the concentration of protein, and you're given 3 marks. This is related to a required practical that you did, and you looked at dilution series of glucose. So this is just looking differently at blood plasma. So you would need, first of all, you'd need to produce known concentrations of protein. You then would measure the absorbance of each concentration, or you could just say measure each concentration with the colorimeter. Then you need to plot a graph. You can't just say plot a graph, you've got to then give a bit more detail for that mark, so of absorbance on the y-axis against concentration, and don't forget you've got to draw the curve, and then you take the sample and then you can find the protein concentration from the curve, and you could explain that you would plot the absorbance and where it intersects with the curve of best fit. You would then draw it down to the x-axis to find the concentration from the curve. And then last question here, it says older people are more likely to suffer from infectious diseases. Suggest how this may be linked to the decrease in the mean concentration of protein in the blood as people get older. So this is related to cell immunity, and very simply that if you have less blood plasma, if you have less proteins in the blood, then you're going to have fewer antibodies. Question 5, and it shows the oxyhemoglobin dissociation curves for fetal hemoglobin and adult hemoglobin. So this is very much related to mass transport in animals, and definitely the role of hemoglobin and loading, transporting and unloading of oxygen. So that is related to 3.3.4.1. In biology, this is a very, very common style of graph related to dissociation curves. So first question, it says explain how changes in the shape of hemoglobin result in the S-shaped sigmoid oxyhemoglobin dissociation curve for HbA, which if we look, HbA was hemoglobin in adult hemoglobin, HbA. So you can see the S-shaped curve there. So we need to explain, not describe the curve, not use any data as such, but you've got to explain. So when the oxygen binds, okay, it causes the change in shape of the hemoglobin, and therefore that change in shape allows more oxygen or O2 to bind easily, or you could say probably better is greater saturation with oxygen. They will also allow cooperative binding. Question 5.2. At birth, 98% of the hemoglobin is HbF, and by the age of 6 months, it has usually completely disappeared from the baby's blood and has been replaced by HbA. Use figure 5 to explain why this change is an advantage for the baby. So you need to look back at figure 5. Okay, there we go. So HbF, you can see, and HbA, well, you can see first of all HbA has a lower affinity for oxygen at low partial pressures. So you can actually see the at lower partial pressure, you lower affinity, and therefore there's easier unloading of oxygen for respiration. Now, easier unloading of oxygen for respiration is important, obviously, as the baby is growing, so lots of tissues, lots of new cells to be supplied with oxygen, so that's really important that there's easier unloading. We then get some form of information. Now, this information is quite applied, and a lot of this information you would not already know. Well, you'd know the basics, but you wouldn't necessarily know the actual example. You may have discussed about sickle cell disease, but don't worry if you haven't, because it tells you it's produced by a faulty HbA. Okay, this results in a reduced ability to transport oxygen to tissues, which is a pretty bad thing because that reduces respiration. So scientists investigated the use of a substance called hydroxyurea, hydroxyurea to treat babies. Hydroxyurea changes the concentration of HbF in the blood. So how does it change it? Well, we're going to have a look. So they carried out the investigation, 122 babies. Each baby was given hydroxyurea for 41 months. And then they found the mean change in the concentration of HbF, because that's what it changes. So you can see before treatment and then after treatment, and they looked at plus and minus 1 standard deviation. So the first one, 7.6, I'm just going to plus 4.5, and I'm going to find that that is 12.1, and the 19.1, I'm going to minus the 6.5, and that gives me 12.6. So I now immediately know that there is a significant difference. There is no overlap of the standard deviations, so it's significant. Will that be useful? We'll wait and see. Question 5.3. The scientists concluded that treatment with hydroxyurea would increase the concentration of oxygen in the blood of babies with SCD. Suggest how Figure 5 and Table 1 support this conclusion. So not only are you going to use the data up above, okay, but you actually, from the other page, you're going to use Figure 5, so the dissociation curves that they showed you. So I'm going to get that now to show you. For 3 marks then, first of all, we've said that there is a large significant increase. There's a significant increase in HbF. That's the first thing to say from Table 1. From Figure 5, we can see that HbF has a higher affinity for oxygen. Okay, that would be, and you can see in the data as well, there's a higher affinity. So the mean concentration of HbF in the baby's blood is higher as well, so there's a higher proportion of HbF in the blood, so more oxygen could be carried, or more oxygen could be carried after treatment. So those are the 3 things that they're looking for. There's 2 answers in the table, so it's significant, and there's a higher proportion of HbF in the blood, so more oxygen is carried. And then looking in the graph, you know that HbF has a higher affinity for oxygen than the faulty HbA. For the 3rd mark point, you could say that more oxygen will be carried after treatment. Question 6, and it talks about stomata. You should know about stomata already, but it tells you where they're found. And they painted a layer of nail varnish on the lower epidermis, peeled off the dry layer of nail varnish, and placed it on a microscope slide. So you've probably done this experiment quite a few times in your biology careers so far. And they looked at the impressions under an optical microscope, so light microscope. Optical light microscope, I should say. She then determined the mean diameter of the stomata. So the first question, describe how the student could use an eyepiece graticule to determine the mean diameter. So this is related to methods of studying cells, so 3.2.1.3. So to measure the diameter of each stomata or stoma, you're going to first use the eyepiece graticule. So the eyepiece graticule is within the eyepiece, and then you calibrate it using the stage micrometer, or you could use ruler or a graph paper to work out how many micrometers you're looking at, and then you take a number of measurements, because if you look at the stem of the question, it does mention you need to determine the mean. So to determine the mean, you have to look at more than 1. So you need to look at a number of stomata. The question gives you a bit more information here. So it talks about an investigation treating the leaves of 1 species of plant with 3 different substances, and they reduce, okay, in bold. If it's in bold, it's important, remember that, the stomatal diameter. He compared the mean diameter, so took many measurements, after treating the leaves with these substances with the mean stomatal diameter on control leaves treated with distilled water. And the results are shown, and it says the mean, this is really important, the mean stomatal diameter of the control was 7.5 micrometers. Right, so if I just show you down here, so we've got on this graph the reduction of mean stomatal diameter compared with the control. So we've got PS3, LAM13 and ABBA, doesn't matter what they are as such, but if I read PS3, that is 0.4. But that's not the figure we're interested in. The figure is compared with the control, so it's 7.5 minus 0.4. So that tells us 7.1 micrometers for PS3. And then ABBA, which is the other one which we're coming up to, okay, or ABA, it's not ABBA. This isn't Waterloo. So 2.7, yeah, get back to it, Dr. B. 2.7, so 7.5 minus 2.7, okay, and that will give you 4.8 micrometers. The question says, calculate, so get your calculators ready, the ratio of the mean stomatal diameter of leaves treated with PS3 to those treated with ABA. So you've got to compare it with the control. So you'll get your 7.5 and you'll divide it by the 4.8 and you get your, and you won't get the answer. It's not 7.5, is it? It's 7.1, sorry. It's that figure there. See, even a genius can get it wrong sometimes. So 7.1 divided by 4.8, and you will get you get 1.479, but I would say round it up, 1.48, and that is the answer. So as I said to you, you just need to read the stem of the question. The reason it's bold, these substances reduce the stomatal diameter, so you've got to work out the reduction of the stomatal diameter, but compare it with the control, which was a 7.5, okay, and that is your answer, 1.48 to 1. 6.3, and it says ABA is a substance that some plant species produce when little water is available, so when there's xerophytes, for example. So explain why producing ABA may help these species survive in dry conditions. So this question is a bit about mass transport, which is 3.3.4.2, and adaptations in plants surviving dry conditions. Now, if you remember from the graph, ABA reduces the stomatal opening. So an explanation for why that is a good thing, well, that will reduce transpiration. There'll be less transpiration, or you could say less evaporation or less water loss by diffusion, but I would say less transpiration. Therefore, okay, how does that help survive in dry conditions? More water is going to be available in the plant for photosynthesis, metabolism, support, or less water will be needed from the dry soil. You can't just say that water would be conserved. You've got to say what the water would be available for in the plant. I think that's the difficult part of this question, the second part. The reason for this is that it does say in the question, explain why producing ABA may help these species survive. If you conserve water, how is that going to allow it to survive? So you've got to go a step further in your thinking. So what is the water going to be used for? And you need to give an example of that. 6.4, many species of plants can be infected by powdery mildew, which is spread by microscopic spores. Powdery mildew is something you may know about, but it's not in the spec. But it then says, suggest how treatment with LAM-13 might protect plants against powdery mildew infection. So the question you need to ask yourself is the microscopic spores in the air. So how are they going to get into the plant? Well, if they're in the air, they're going to enter through the stomata. So LAM-13, how does it protect it? Well, no spores will be able to enter the leaf because the stomata close. This question is pretty, well, it's a lot applied, basically. A lot applied, is that a statement? No, I don't think it is. Anyway, but you can see, and I keep on going on about this a lot with my own students. Look how much information they've given you in that question. They've told you that powdery mildew is spread in the air by microscopic spores. So in terms of being infected, how is it going to get in? Well, it's going to get in through the stomata. So, if you treat it with LAM13, looking at the graph, you'll be able to see that LAM13, the stomata close, or there's fewer stomata open, and therefore no spores can enter that leaf. Notice spores is underlined. Alright, that is for a reason, is because you need to mention spores in your answer. On to question 7.1, and it tells you NMO is a disease that leads to damage to nerve cells in the spinal cord. Again, NMO is not in the spec, alright, but it tells you what it does. A person with NMO produces anti-AQP4 antibody. Now in that statement, anti-AQP4, I'm pretty certain you probably won't know what that is, but you all should know what an antibody is. And it attacks only these nerve cells, so it's specific to the NMO, the damaged nerve cells. It says explain why the anti-AQP4 antibody only damages these cells, and it's for 4 marks. In the spec, this is related to cell recognition and the immune system. Now, this is not related to responses, humoral response to immunity or anything like that. It's related to antibodies, and it's related to the formation of antigen-antibody complexes, and you need to talk about the specific nature of what exactly is happening. So you're going to get a full 4 marks for this. Now, again, in the stem of the question, it just says antibody. It doesn't actually tell you what an antibody is or its structure, so you can use that as an explanation. So if we remember about the antibody, it has a variable region and it has a particular Y shape, and you can see that it has binding sites. It has a tertiary structure and it's got binding sites, and those binding sites in the variable region, they are complementary to one type of antigen. So let's say I'm going to do this one particular antigen, and that antigen fits in, or in fact the antibody moves towards the antigen, and the antigen to this antibody are found on this horrible NMO. There we go. So the NMO antigens are that shape. So, and they're only found on those nerve cells. So the antibody binds to form an antigen-antibody complex. If you're going to remember 3 words related to immunity, related to antibodies, antigen-antibody complex with these particular NMOs, nerve cells. Let's cover up my terrible illustration. So yeah, here we go. So exactly what I've just said, basically. Key thing to remember, okay, they're not active sites. You don't talk about active sites, you talk about binding sites when it's about antibodies. They also, as I said, have a specific tertiary structure. The antigen, if you're going to mention, you can mention receptor for antigen, but I would mention antigen, or you could say antigen receptors that bind to the to form an antigen antibody complex. And then common theme in these types of questions, they then give you some further data and information, and it talks about a scientist doing an experiment looking at the concentration of anti-AQP4 antibody in the blood of people with NMO, and the spinal cord is surrounded by small bones called the vertebrae or vertebra. For each person, the scientists also determine the number of vertebrae surrounding damaged nerve cells. Okay. And they show the results in this handy scatter graph. So you can see the number of vertebrae surrounding damaged nerve cells and the concentration of anti-AQP4 antibody. Now, you can see quite a weak correlation, but there's definitely some form of correlation there, with quite a few outliers. But you can see generally as the number of vertebrae surrounding the damaged nerve cells increases, so does the concentration of anti-AQP4 antibody. However, that's not the question they're going to ask for this one anyway. The scientists suggested that the concentration of anti-AQP4 antibody in a person's blood could be used to predict the number of vertebrae surrounding damaged nerve cells they are likely to have. And it says, use figure 7. Oh, look, it's bold again. Use it to suggest reasons why this suggestion might not be valid. Right, so this is about scientific methodology. The first thing to note is they say, for each person, the scientists determine the number of vertebrae surrounding the damaged nerve cells. Now, they don't tell you the number of people, but if I count the number of dots, okay, I can count those dots, all those dots, I'm going to get the figure of 20. Now, 20 people in a study is not a lot, so it's only 20 in the study, or it's a small sample. It's also only one study as well, so it's not multiple studies, so that's something to think about. They haven't done any form of statistical test, they've just shown the raw data. As I said to you before, the correlation is there, but it is very weak. The correlation is very weak because there's lots of outliers around the line of best fit. Also, if you look at some concentrations of anti-AQP4 antibody, there is a range in the number of vertebrae surrounding damaged nerve cells. So, for example, at this point here. So, it's probably, those are the kinds of reasons that they're looking at, why it's not valid. And notice all those points are there in the information. So you don't need to know you need to know about scientific methodology, but really you can answer that by just analysing the stem of the question and the data that they give you. So there's the mark scheme. You only need 3 of those points to get all 3 marks, just from looking at the information given to you in figure 7. 7.3 then goes on to say a new treatment for NMO involves using a monoclonal antibody. Now, you'll know that's in your spec. You need to know about monoclonal antibodies. And it says the structure of the variable region is identical to the variable region of an anti-AQP4 antibody, but the rest is different, the rest of the structure. Use this information and your knowledge of antigen-antibody complexes to suggest how this monoclonal antibody prevents anti-AQP4 damaging the nerve cells. Only just 2 marks, all that information, and they want 2 marks worth. So the key thing is to think, well, this is related to competitive inhibitors, but you're not going to be saying it's a competitive inhibitor. What they want to know is how it prevents anti-AQP4 damaging the nerve cell. So what will the monoclonal antibody be doing? Well, it will bind to the nerve cell antigen. So if it binds to the nerve cell antigen, less or no anti-AQP4 can bind. And when it binds, when the monoclonal antibody binds, it doesn't cause damage to the nerve cell. Now, in your answer, you can say it, meaning monoclonal antibody. Again, don't talk about active sites, competitive inhibitors, or enzymes. That's not what it's about. The monoclonal antibody is binding to the nerve cell, so less anti-AQP4 can bind, but it's not necessarily a competitive inhibitor in the correct sense of the word related to enzyme substrate complexes, so it's slightly different, but it is competing for the antigen to bind with the nerve cell antigen. So when it binds, it doesn't cause damage to the nerve cell, so that is why that second part's important, because that's how it prevents the anti-AQP4 damaging the nerve cell. Question 8, and it says Patau syndrome, which is not in your spec, is a condition caused by mutation affecting chromosome number. Mutations is definitely in your spec. Okay, so all the cells of the body will have this mutation. Shows the chromosomes of a female with Patau syndrome. You can see it's a female because it's XY there. So let's have a look at the question. What is the effect of Patau syndrome on the chromosomes of this female? So they show you all the chromosomes, and you can see the homologous pairs for 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. I'd say there was a problem there. Extra chromosome. So it says, what is the effect on the chromosomes? So you will have to say there are 3 of chromosome 13. So 3 of chromosome 13, or you could say there's an extra chromosome on chromosome 13. Got to mention chromosome 13 though for the answer. So 8.2 and also 8.1 is related to 3.4.3 in the spec, which is related to genetic diversity can arise as a result of mutation or during meiosis. And the question says describe, and I've highlighted describe, which is great. Describe is recall how the change in chromosome number was produced. Now, Patau syndrome, all you need to know is that an extra chromosome has been produced. There has been a mutation in the number of chromosomes. And it says, how was it produced? Now, this is directly related to the spec, okay, because in the spec it says mutations in the number of chromosomes can arise spontaneously by chromosome non-disjunction during meiosis. So meiosis is not mentioned in the answers, so that's the first one. And you can say that the homologous chromosomes do not separate, or non-disjunction. The reason they'll allow that is because I'm looking at it now in the spec, it's directly mentioned in the spec. So if you've learnt meiosis and the fact that they arise spontaneously by chromosome non-disjunction, then that is the answer. Question 8.3, it says explain why all the cells of the body will have this mutation. So it's not going to be meiosis, it's going to be related to mitosis in this respect. So there's 2 marks, there's quite a lot of things you could say. So 8.3, so you can say the chromosome is in the gamete, for example, that forms a zygote, or all cells are derived by mitosis, which is not mentioned in the stem of the question. Or you could say all cells derived from a single cell by mitosis, and also something you'll be aware of, that mitosis produces genetically identical cells. Again, it goes on to give some further information about Patau syndrome, and it tells you that unfortunately most children born with this syndrome die in the first 12 months, often due to defects of circulation of the blood. So circulation of blood is related to mass transport in animals. So now we're looking at spec 3.3.4.1. It says one of these defects is patent ductus arteriosus, or SUS, which is PDA. Again, would you know that? No, you would not. However, tells you what it does, and it results in some of the blood flowing between the aorta and the pulmonary artery, which is bad news. It shows, and figure 9 shows a healthy child's heart and then a child's heart with PDA, showing, as you can see, the movement of blood from the aorta. Oop, is it not working? It's not working. There we go, aorta into the pulmonary artery. So, question. It says, suggest how the flow of some of the blood between the aorta and pulmonary artery could cause children to die in the first 12 months. So key things there, suggest, so there's quite a few answers, and you've got to talk about why that is going to be fatal for children under 12 months. So for this question, you're going to need to understand the growth structure and function of the heart, and you'll need to know that the aorta on the left side has oxygenated blood, and it will flow into the pulmonary artery, and the pulmonary artery is then going to the lungs. Now, that in itself is not the issue. However, the issue is that if the aorta is leaking oxygenated blood, then it's not going to go into the left ventricle, and it's not going to then travel through the aorta to deliver oxygen to the cells. If it can't do that, then, and this is kind of stock answer, less oxygen, therefore not enough for oxygen for respiration, therefore the organ doesn't grow or develop properly. Now, why that last question? Well, you've got to remember it's about children in the first 12 months of life. So if that organ, if the heart does not grow properly or other organs don't, that will cause death. So there's the answers they would accept. They won't accept things like saying no oxygen, because some oxygen is going to get through. And definitely, I've talked about this about a million times, they do not accept produce energy. They don't produce energy. You release energy from respiration. Key one that people might not get is that the organ doesn't grow or develop, so therefore that's what is causing death in the young children, which is very sad indeed, actually. On to the last question, and again in paper 2 there will be 2 5-mark questions. If you have done your revision, usually you can pick up most, if not all, of these types of marks, and you can go up a whole grade boundary with 10 marks. So it says, describe the cohesion tension theory of water transport in the xylem. So immediately you think it's mass transport of plants, cohesion tension theory, and off you go. So mass transport in plants, I'm just looking at the spec, that is 3.3.4.2. Let's have a look at the types of answers that they would accept. So for 5 marks, you've got to mention water is lost from the leaf because of transpiration. They would allow evaporation of water or diffusion from the mesophyll or leaf cells. But you can't just say water is lost. All right, you can't say water is lost through the stomata, for example. It's because of transpiration. That's the first one. Or you could say transpiration through the stomata. If you were going to say water is lost through the stomata, you need to say diffusion of water. So what does that do? Well, it lowers the water potential of the mesophyll cells. That then means that water is pulled up the xylem, and I would say creating tension, and then they cohere. So water molecules are cohesive, they stick together by hydrogen bonds, and it causes a continuous water column. And then you get adhesion as well. This last one, you could mention it, not just adhesion of water molecules, cohesion, sorry, not adhesion, cohesion, but water adhesion occurs on the walls of the xylem. So that kind of question, if you've done your revision, is, you should be able to do quite well. If you haven't done your revision, then you need to think about the facts that you probably know it anyway, but cohesion and tension. So what's causing the tension? Well, it's the fact that water is leaving the leaf because of transpiration, or leaving the leaf through diffusion of water through the stomata, and cohesion because water molecules are cohesive and they stick together and they're going to be moving up the plant. So you could get some of those marks. Then on to the final question, and again, describe how mRNA is produced in a plant cell. So mRNA, how is it produced? Doesn't matter whether it's a plant cell, it could be an animal cell, but we're talking about protein synthesis, which is 3.4.2. Now, in terms of this, another 5 marks, so it tells you nothing about the production, how it's produced. It's going to be related to translation. Okay, so you would need to talk about the DNA being, the bonds being hydrolyzed or broken. One of the strands acts as a template to make mRNA, and you could talk about base pairing, so A to U, for example. You could then say the nucleotides are joined by RNA polymerase. Pre-mRNA is formed. Remember, it's pre-mRNA inside the nucleus, and then for it to move into the cytoplasm, it has to be spliced so the introns are removed. So you've got a lot of different things you can talk about there. So there is the mark scheme. It also will accept, for mark point 1, it accepts DNA unzips by breaking the hydrogen bonds. It does say ignore hydrolysis of bonds, so they want you to say it breaks the hydrogen bonds. But there we go, so transcription, that's all you would need to talk about. I hope you found that run through useful. Okay, so please do subscribe to Dr. Biology. I'd be eternally grateful. In terms of the A-level papers coming up, I'm going to do quite in a short while the AS Biology Paper 1 and Paper 2 from 2018. So that is something to look out for. Last year's AS paper. So take care. And have a good one, and keep up with your biology. I know you can do it.